Welcome back, everyone. In our last discussion about carbohydrate metabolism, we had completed the glycolytic pathway, but I had actually missed out one small detail about the glycolysis, and that is the very first thing that we are about to discuss in this discussion. Okay, then we will move on. Let me tell you about the scope of this discussion today. Then we will move on to the metabolism of fructose galactose, and we will also see briefly the sorbitol pathway, and then we will finish off the discussion with a brief discussion about the gluconeogenesis. Very easy. Okay, it's very easy. Everything is very easy today. So we have completed glycolysis, but I have to mention rather just one small thing. Okay. That one small thing is, if you remember, in glycolysis we had this step where fructose 1,6-bisphosphate was being cleaved into by aldolase A, by the way, into what we say to be glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, right? That glyceraldehyde 3-phosphate, that was being converted into 1,3-bisphosphoglycerate, right? 1,3 this was 1,3-bisphosphoglycerate, 1,3-BPG. This was one of the steps, right? And obviously we know that this is one of the only steps where reduction is occurring and NAD plus is being converted to NADH plus H plus. And since this is dehydrogenation, so the name of the enzyme is glyceraldehyde 3-phosphate dehydrogenase. Glyceraldehyde 3-phosphate, I'm abbreviating it, dehydrogenase. This is something that we already know about. The next step which used to occur is 1,3-bisphosphoglycerate being converted to what we say to be 3-phosphoglycerate, right? This was another step, and this is one of those steps where ATP was formed, because this is the 7th step. So ADP to ATP, this happened. The name of the enzyme which did this was phosphoglycerate kinase. Okay, phosphoglycerate kinase. Now this entire thing we know. What we don't know is what happens in some specialized cells like erythrocytes. What happens? See, in erythrocytes, 1,3-BPG is not directly converted into 3-PG. Rather, 1,3-BPG is converted into what we say to be 2,3-BPG. 1,3-bisphosphoglycerate is converted by the enzyme bisphosphoglycerate mutase into 2,3-bisphosphoglycerate. This 2,3-bisphosphoglycerate by the enzyme bisphosphoglycerate phosphatase. This enzyme converts the 2,3-BPG back into 3-phosphoglycerate. So in erythrocytes, normally what happens is directly this conversion does not take place, rather this conversion takes place by this process. Why? Why exactly does this happen, and why is it so important for us to know this? The reason for this is 2,3-BPG is a very special compound. It's a very special molecule, and in high school biology we have learned a little bit about this when we studied about respiration. You see, 2,3-BPG is such a molecule which will decrease the affinity of oxygen for hemoglobin, or rather decrease the affinity of oxygen for hemoglobin, or of hemoglobin for oxygen rather. That's the grammatically correct thing. So you're getting the picture, right? Like the hemoglobin HbA I'm talking about, mostly because this is adult hemoglobin that we talk about, that has a certain affinity for oxygen, obviously. What 2,3-BPG will do is it will bind with the hemoglobin A, and it will decrease oxygen affinity. Okay, mind the term. Hence, what will happen, how is this beneficial, or why that is important, is because if a hemoglobin molecule's affinity for oxygen decreases, then that hemoglobin molecule will easily give up that oxygen molecule, right? How is that important? That is important because easily giving up oxygen would mean that the hemoglobin would deliver oxygen more better, or not better rather, not more better, because it will easily give up the oxygen to the needy tissues. If the hemoglobin is to hold the oxygen for itself, then the function will be compromised. But if its affinity is decreased, then it will easily deliver the oxygen to the tissues which need it. And this is exactly what happens when we are in hypoxic conditions. Okay, so in hypoxic conditions, that is where oxygen level is less, we are in a dire need for oxygen, such as in higher altitudes. There our body automatically adapts to the situation by increasing 2,3-BPG concentration in the blood. Hence, this 2,3-BPG, what that will do is it will decrease oxygen affinity for hemoglobin molecules. So the hemoglobin molecules will automatically be better at delivering the bound oxygen to the needing tissues. Hence, 2,3-BPG is a very, very important molecule. Thus, 2,3-BPG is how that is produced. That is produced from this pathway, or this, you may say, shunt. Why this is a shunt? Because this was the normal pathway, and now you're shunting 1,3-BPG not by the normal but by a bypass pathway. That's why this is known as a shunt, or namely this shunt is known as what we say to be Rappaport-Louis Behring shunt. Okay, Rappaport-Louis Behring shunt. Note the spelling, L-U-E-B-E-R-I-N-G. Okay, Rappaport-Louis Behring shunt, or also known as the BPG shunt. Okay, Rappaport-Louis Behring shunt or the BPG shunt. So why this is important, you get it? Because 2,3-BPG is needed for erythrocytes. Hence, erythrocytes go through this special pathway so that the 2,3-BPG can be synthesized, which in turn regulates oxygen affinity for hemoglobin molecules, which is an extremely important task. Okay, so this is an important thing which I forgot to mention in my previous video. Now we move on to today's scope. Today's scope is about the metabolism of the other monosaccharides, that is fructose and galactose. Okay, so let's first talk about the fructose metabolism. It's very easy. It's nowhere near as comprehensive as glucose metabolism, right? So fructose. Okay, now listen to me very carefully. We do get fructose from our diet. That's easy, that's obviously correct. For example, we eat fruits. Fruits are very rich in disaccharides like sucrose, and sucrose obviously has fructose in them, right? So fructose is something that we obviously encounter in our diet, right? Now it's also present in sweets, sugar, right? So fructose, once it is I would say digested, that is the sucrose or anything, whatever the complex source is, it's broken down, and fructose we get it in our gut. After that occurs, what is needed then? Fructose needs to be absorbed by our body. From the gut, by the epithelial cells, fructose needs to be absorbed. How that occurs? That occurs by facilitated diffusion. Okay, so fructose enters the epithelial cells by facilitated diffusion, and mostly, this is a very important piece of information, fructose is mostly transported by GLUT5. I think I had mentioned this before. GLUT5 is that one glucose transporter, although this is a glucose transporter, but GLUT5 has a higher affinity for fructose than glucose. So GLUT5 is an important transporter of fructose, and overall fructose actually is transported by facilitated diffusion. Now fructose, what's the importance of fructose in our body? Let me tell you that first. Fructose is the primary source, the primary 1 degree primary source of energy, not for cells but for sperm cells. Okay, specifically the sperm cells, their primary source of energy is fructose. So fructose is important, right? Right. Now let's see, after the absorption takes place, after fructose has entered into the cells of our body, how our body will utilize fructose. This is the story of, or rather this is where the metabolism of fructose starts. Let's see to that. See, fructose, we have fructose. What at the very beginning what happens is fructose will be phosphorylated, and what is formed is fructose 1-phosphate. Very important. Fructose will be phosphorylated and the formed product is fructose 1-phosphate. So what's the enzyme needed here? Obviously it's a kinase. What kinase? If you remember, in case of glucose, the name of the kinase was either glucokinase or hexokinase, right? Similarly, here the name of the kinase is fructokinase or hexokinase. Interesting. You see, hexokinase, hexose sugar. So this is a common kinase, and fructokinase is more specific for fructose, just like glucokinase was more specific for glucose, but hexokinase is common in both. And in fact, let me break the suspense right here, in galactose also hexokinase is a phosphorylating enzyme apart from galactokinase. So all the three monosaccharides follow the same trend of phosphorylation at the beginning of their metabolism, and their phosphorylating enzymes also are very similar. Glucose will be phosphorylated by glucokinase or hexokinase, fructose will be phosphorylated by fructokinase or hexokinase, and galactose will be phosphorylated by galactokinase or hexokinase again. So hexokinase is common for all the three, right? Right. So fructose 1-phosphate is formed here. Now this is where the interesting stuff occurs. This fructose 1-phosphate will be directly split. It will be split into what we say to be glyceraldehyde only and dihydroxyacetone phosphate. Now listen to me very carefully. If you remember glucose metabolism, in glucose metabolism what had we formed? We had formed from glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, and then we had split it, right? And we had got glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So there is some similarity between these two, but the difference is there too. Listen to me very carefully. See, in fructose, the phosphorylation is occurring and it is forming fructose 1-phosphate. If it had formed fructose 6-phosphate, then from there onwards we would have followed the same glycolytic pathway, but that does not occur. Are you getting me? Fructose is phosphorylated by fructokinase or hexokinase. In fact, mostly by fructokinase. I'll tell you about the story of hexokinase a little bit later. So by fructokinase into fructose 1-phosphate. If it had been fructose 6-phosphate, then the story would have ended right there. Right there. There wouldn't have been anything else to talk about pertaining to the speciality of fructose metabolism. Because if this had been fructose 6-phosphate, the same story would have continued, but that does not occur. What does occur is fructose 1-phosphate is formed, and this fructose 1-phosphate is not phosphorylated further into fructose 1,6-bisphosphate. No. This fructose 1-phosphate is directly split. Okay, so it is directly split. And see, in glucose metabolism, it was fructose 1,6-bisphosphate which had split, and hence both the products which were formed had 1 phosphate each. But in this case, there is only 1 phosphate at the beginning, fructose 1-phosphate. Only 1 phosphate is attached to the first carbon, right? So in the split products, only one of the product has a phosphate and the other one does not have. Okay, so this is the sharp contrast between the fructose metabolism and glucose metabolism. Okay. By the way, another very similar thing is, what was the enzyme which actually catalyzed this splitting up? Fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. What was the name of the enzyme? Yes, very good. Aldolase. But do you remember I had said that aldolase has some isoenzymatic forms, out of which A and B are the most common, right? This aldolase, the isoenzymatic form of aldolase in this case is aldolase A. Aldolase A. Whereas in this case it is aldolase B. Very, very important information. In glucose metabolism, fructose 1,6-bisphosphate was split into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by the action of aldolase A, the A isoenzyme form of aldolase. In fructose metabolism, fructose 1-phosphate is split by aldolase only. It is aldolase. In both cases, it is aldolase which does the splitting. But in glucose, where fructose 1,6-bisphosphate was being split, it was aldolase A which was doing the work. And in fructose metabolism, where fructose 1-phosphate is being split, it is aldolase B which is doing the work. So very, very important information. Okay. These two enzymes, fructokinase and aldolase B, these are very important enzymes because clinical relevance lies with these two enzymes. Hence, they are extremely important, and we will talk about this in a few minutes. So, here is the thing. See, in both fructose metabolism and galactose metabolism, Why are we studying this separately? We are studying them separately only to see how special they are in contrast to glucose metabolism or glycolysis in this case. We are talking about the first, that is glycolysis. The speciality of either fructose or galactose metabolism ends when they form one of the intermediates of glycolysis. Because if they form even any one of the intermediates, from that intermediate onwards glycolysis will continue. Prior to that intermediate, what occurs? That is the thing that I am trying to talk about. So have we caught on to any of the intermediates yet? Yes, we have. We have caught on to dihydroxyacetone phosphate, but glyceraldehyde is not yet converted to glyceraldehyde 3-phosphate. So we do need to convert glyceraldehyde into glyceraldehyde 3-phosphate, right? So the next step is pretty self-explanatory. Glyceraldehyde is phosphorylated to form glyceraldehyde 3-phosphate, and obviously an ATP is phosphorylated. An ATP is utilized here as the phosphate donor. Okay. By the way, the name of this enzyme is triose kinase. Okay, the name of this enzyme, triose, because this is a 3-carbon compound. Glyceraldehyde is a triose, that is a 3-carbon sugar. Okay, so and kinase is phosphorylation occurring, so for kinase. So triose kinase is the name of the enzyme which will phosphorylate glyceraldehyde to glyceraldehyde 3-phosphate, and from here onwards it is essentially the same. See? So that's what I'm trying to explain. The story of fructose metabolism or galactose metabolism ends when any one of the intermediate of glycolysis is formed. After that is formed, from there onwards normal glycolysis will go on. So from here on, normal glycolysis. Okay, so that's that about fructose metabolism. So this is how fructose metabolism occurs. Okay. Now let's talk a little bit more about this and how the clinical relevance lies with these steps. Let's talk about that now. So here is the thing. You see, I said a thing about fructokinase and hexokinase. There is a difference between them. Okay, but the thing is, hexokinase mostly works in fructose metabolism. Hexokinase mostly works when fructose level is quite high. So when fructose level is quite high in blood, that is when hexokinase will work. Otherwise it will be the work of fructokinase to phosphorylate fructose. Okay, that's an important information. And there is another thing, let me tell you. See, so hexokinase works when fructose concentration is very high. There is another case where hexokinase will work, where it will work when fructokinase is deficient. So these are the two conditions when hexokinase will work. Either the fructose concentration in blood is quite high, so hexokinase needs to kick in, or fructokinase, which is the normal enzyme, which is the common enzyme, if that has a deficiency, if there is a deficiency of fructokinase, then hexokinase will work. How this is clinically relevant is because, see, if fructokinase is deficient, the condition is known as what we say to be essential fructokinase fructosuria. Essential fructosuria. So fructosuria means there is excessive fructose in blood. Okay, so the condition where fructokinase, this enzyme is deficient, the condition that will be prevalent then will be essential fructosuria. So in case of essential fructosuria, what happens? Let's talk about that. So now we have already blended into the clinical part of fructose metabolism. Okay. So this is the first clinical part. Now let's talk about essential fructosuria. See, essential fructosuria, as you can all pretty much guess by this point, since this is a enzyme deficiency problem, when can fructokinase be deficient? Think about this. Fructokinase is an enzyme, so it's a protein. So it's obviously it has a gene which is encoding the information for this enzyme. If there is a genetic problem, that is, if there is a problem in that gene, then fructokinase will be deficient. And this is the story for most enzymatic problems, right? That is genetic problem. Okay, so if this is genetic, we can say that is this autosomal? In case of inheritance, is this autosomal or is this sex linked? The answer is this is autosomal. Autosomal. So this disorder is autosomal recessive. Okay, recessive. So it's not dominant, it's an autosomal recessive disorder. The second point which I would like to say about essential fructosuria is, see, in case of essential fructosuria, what will happen is fructose will start accumulating, right? But I have already said that hexokinase works when fructokinase is deficient. So hexokinase will make up, mostly, it will make up for the fructokinase deficiency. Hence, since hexokinase will make up for that, hence essential fructosuria is a benign, I think this is the benign is, yeah, benign and asymptomatic condition. See, logical, it's very logical. There's nothing to mug up here. I said the first thing is that this is genetic. So if this is genetic, then there is the possibility of this being inherited. So in that case, this is autosomal recessive. It's an autosomal recessive disorder. Okay. Now I have already said that fructokinase, if that is deficient, hexokinase will make up for that mostly. So hexokinase will make up for the deficient fructokinase. So since this was a problem for fructokinase deficiency, but you have already found a supplement, so there are almost no symptoms and quite normal, benign asymptomatic condition. Okay, and mostly also what happens is that the excess fructose, if present, if the excess fructose is present rather, then obviously just like glucose, that excess fructose is excreted out through urine. Okay, so obviously this is just like hyperglycemia, except that this is essential fructosuria where fructose levels are quite high. Okay. So this is the first clinical condition regarding fructose metabolism, which is fructokinase deficiency. Now if you remember, there was another important enzyme in fructose metabolism. What was that? Yes, aldolase B. Aldolase B was another enzyme. So if aldolase B is deficient, then there is another condition which is known as fructose intolerance. Fructose intolerance, or rather hereditary fructose intolerance. So what about hereditary fructose intolerance? Hereditary fructose intolerance is a little more serious condition than essential fructosuria. Obviously that occurs when aldolase B is deficient. So if aldolase B is deficient, that would mean that fructose 1-phosphate would start increasing, right? Fructose 1-phosphate would start increasing. Now fructose 1-phosphate, this mostly is accumulated where? In hepatocytes, in the liver, right? So due to excessive fructose 1-phosphate accumulation, what may occur is hepatocytes may start dying. Very, very morbid. This is a grim detail, right? That fructose 1-phosphate, which is essentially not very, I would say, body friendly, it's actually a little toxic. It's a toxic intermediate of fructose. If that starts accumulating due to aldolase B deficiency, the hepatocytes where they are accumulating would start dying. So there is rapid death of hepatocytes. Now, if there are hepatocytes number is decreasing, that would lead to jaundice. So jaundice is another, we may say, condition or rather symptom which is associated with essential fructose, rather hereditary fructose intolerance. Another condition is, not condition, the symptom is, logically, let's think about this logically. If fructose 1-phosphate is accumulating, obviously that's a carbohydrate metabolite, right? That's a carbohydrate intermediate. So the body would itself inhibit its production of any other monosaccharide or carbohydrate, which would mean that glucose production will be inhibited by body, which would in turn lead to hypoglycemia. Very important. So hereditary fructose intolerance would in turn lead to hypoglycemia because you already have carbohydrate intermediates building up. You don't want any more carbohydrate sources. So glucose production in body would decrease, would be stopped rather, and hypoglycemia would follow. Okay. And the other more physical symptoms you may say would be obviously vomiting will be there, convulsions will be there. Okay. These are some of the other symptoms which are associated. However, there is another thing. First thing is that just like the fructokinase deficiency, aldolase B deficiency is also what we say to be autosomal recessive condition. So this is also autosomal recessive. Okay, this is also autosomal recessive condition. And let me tell you another thing. Just like in case of fructose or rather fructokinase, we said that it is asymptomatic condition, right? In hereditary fructose intolerance also, this is actually what we say to be asymptomatic. Asymptomatic. Then what are these? These were all symptoms. These are all symptoms. So many symptoms I just jotted down, and then I'm saying that this is asymptomatic. Wait, I haven't finished it. Asymptomatic until fructose is introduced in body. This is where the detail lies. So the devil is in the details. Asymptomatic until and unless fructose is introduced in the body. So the best treatment for this is obviously to completely stay away from any fructose sources in diet, because if fructose is introduced in body, that is when these conditions will start kicking in. Otherwise asymptomatic. Okay. So that finishes off the I would say fructose metabolism. Okay, now let's move on to another thing. Okay, since we are talking about fructose, let's talk about another thing. Listen to me carefully. See, just like we said that fructose is it's an essential part of our diet because it's found in fruits, sweets, etc. But fructose is also produced in our body. It is also produced in our body. From what? From glucose. From glucose, fructose is produced in our body also by a pathway which is known as the sorbitol pathway. The sorbitol pathway. Now, nothing to be paranoid about. This is just a single step, or rather 2 steps actually. See, let me tell you. The aim of this pathway is to produce fructose from glucose. Why this is important? Because this has some clinical relevance. Very, I would say very important clinical relevance, and I'll tell you why it's worth remembering this pathway. Okay? Okay. Glucose, that's present. Okay? Glucose is converted into the name of this sorbitol. Sorbitol. Now, what I like to think of sorbitol is the alcoholic form of glucose. See, glucose is the aldehyde, right? So, an aldehyde is being converted to its alcohol form. For example, if I said that ethanol or acetaldehyde is being converted to ethyl alcohol, just like that, glucose is being converted to sorbitol. So it's the alcohol form of the aldehyde, that is glucose. So glucose is aldehyde, sorbitol is alcohol. So now from basic organic chemistry knowledge, we know that aldehyde to alcohol would be essentially reduction, because acid to aldehyde is reduction, and from aldehyde to alcohol is reduction, and the other way around will be oxidation. Okay, basic organic chemistry, high school. So glucose to sorbitol, this would essentially be what? Reduction. So you need again what? Something to accept the, or something to actually give the reducing equivalence, right? That something is none other than NADPH plus H plus. NADPH, see, till now in glycolysis we had studied about NADH, not NADPH, but in sorbitol pathway we have NADPH plus H plus. This NADPH plus H plus would donate its reducing equivalence, hence reducing glucose to sorbitol and itself being converted into NADP plus. Okay, NADP plus. Now, obviously we need an enzyme here. The name of the enzyme is, I actually have devised a mnemonic for this. So let's see the next step, which is the last step, and then we will name both the enzymes. See, sorbitol is then converted into what we say to be fructose. As simple as that. This is the sorbitol pathway, nothing. So sorbitol is then converted into fructose, but here also there is what we say to be reduction. NADPH plus H plus to NADP plus. Here also there is reduction. Now, about the name of these two enzymes, I actually have devised a mnemonic which is already so dehydrated. Already so dehydrated. Somebody is asking someone that you are already so dehydrated. So this is what the name of the two enzymes are. Already. This is the name of the first enzyme. So dehydrated is the name of the second enzyme. No, these are not the literal names. These represent the mnemonic. So all, L, or rather let me write it here. aldose. Ready, R-E, or rather you may also think it to be R-E-D, red, ready, not R-E-A-D as the spelling suggests, but the pronunciation suggests red, right? So red would be reductase, aldose reductase. So dehydrated, so is this sorbitol, dehydrated, dehydrogenase, okay. So aldose reductase already so dehydrated, sorbitol dehydrogenase, not dehydratase, because there is no dehydration going on, so that's common sense. This is just common redox reaction, so the name is dehydrogenase. That should ring in your mind. Okay, so that is sorbitol dehydrogenase. One very important detail that I would like to present here is, see, aldose reductase, this enzyme is not present in liver. This is not present in liver, whereas sorbitol dehydrogenase, this enzyme is present in liver. Okay, this is an important information, I think. Okay, now where does the sorbitol pathway mostly take place? The sorbitol pathway actually mostly takes place in, as I have said, fructose is the primary source of energy for, yes, sperms. So sorbitol pathway mostly takes place in spermatozoa, right? Also takes place in the lens, eye, in our eye, the lens, and retina. Okay, how this information is very important is where the clinical relevance of sorbitol pathway lies. Now let me tell you about the clinical relevance of sorbitol pathway. Very, very important. You know, in case of diabetics, people who have diabetics mostly they have a high risk of cataract, right? They have a very high risk of cataract, that is losing their eyesight. Why is that the case? That is the case mostly because of the sorbitol pathway. Interesting? Let's see how. You see, I said that the sorbitol pathway occurs largely in spermatozoa and also in the lens and retina cells, right? Now, if a patient is diabetic, then obviously blood glucose level is very high. Now, if blood glucose level is very high, then obviously a lot of glucose will be channeled to the sorbitol pathway, right? So if there is a lot of glucose being channeled to sorbitol pathway, there will be a lot of sorbitol being formed in the first place, right? A lot of sorbitol would be formed. Now, here is a twist. You see, sorbitol actually cannot pass the cell membrane. Very important piece of information. Sorbitol cannot pass the cell membrane. So what will happen? What happens is, since sorbitol cannot pass the cell membrane and since there is a lot of sorbitol being formed, so sorbitol will start accumulating. Where will it start accumulating? Obviously in the places where it is being formed in the first place. Where is it being formed? Lens, retina. So in lens or retina, the extra and excess sorbitol will start accumulating, hence leading to eye problems like cataract or glaucoma. Also, sorbitol also accumulates in not only the retina but also in the kidney, okay, in the nephrons. In the nephrons also it will start accumulating. In the I would say nerves, also it will start accumulating. Hence, excess sorbitol due to diabetes, excess sorbitol will be produced, and since in these places also sorbitol is accumulating, in diabetic persons, obviously some neurological or neuropathic problems, due to this, not this, and some obviously vascular and urinary problems are seen. Okay, so this is the entire reason why sorbitol pathway is so important to know. Okay, so in diabetics, they have the chance of, they have a high risk of cataract, glaucoma, eye problems, neurological problems, neuropathic problems, urinary problems, vascular problems also, only because of the accumulation of sorbitol like compounds which cannot cross cell membrane and hence accumulate. Okay, so to combat this, what is actually being developed nowadays is aldose reductase inhibitor. So inhibitors for this enzyme aldose reductase are being developed. What these will do is they will obviously inhibit this, so they will inhibit the sorbitol pathway, and so sorbitol would be not formed and hence will not be able to accumulate even in diabetic persons. Right. So that's about the sorbitol pathway. Now let's quickly talk about the galactose metabolism, because galactose metabolism also has some very important clinical correlations, and the clinical correlations are the reasons why we are studying them at the first place. So let's talk about briefly, obviously, the galactose metabolism. Now, what is the biggest source of galactose in our body? Obviously diet. What is the biggest source of galactose in diet? The answer is lactose, milk sources. Okay, so lactose, what's actually happening is lactose is broken down by this enzyme, very common enzyme, and I have already mentioned this in my first discussion about carbohydrate metabolism, beta galactosidase into galactose and glucose, but we are not writing down glucose here because we are interested in only galactose. Okay, so this, what I am about to write here right now, is what happens to galactose in our body, how that is broken down, how at the end, what did I say previously when I was talking about fructose metabolism? The story ends when any one of the glycolytic intermediates are formed, because from there onwards normal glycolysis. So when galactose will form something which is a path of glycolytic pathway, the story will end there. So let's see how that happens and what is formed. Right. Okay, now the first thing that or rather not the first thing, there is no first or second or third things. One of the things which can be a fate of galactose is, sorry for the blur. Yeah, so one of the things that can happen to galactose is it can be reduced then and there itself. Okay, see, we had just now said that in the sorbitol pathway that glucose being reduced by aldose reductase would give its alcoholic form, which is sorbitol. Just like that, galactose may also encounter a fate like that. Hence, by aldose reductase, okay, and form its alcoholic form, the alcohol corresponding to galactose, which is galactitol. Right, this compound, this is the alcoholic form of, like, galactose is obviously the aldehyde form and the alcohol form is galactitol via the enzyme aldose reductase, and obviously since this is reductase, so there will be a reduction needed, and the reducing equivalents are provided just like the sorbitol pathway by NADPH, not NADH, but NADPH plus H plus. And hence itself being converted into NADP plus and reducing the galactose into galactitol. This is one of the fates of galactose, but only one of the fates. But this is not the more common fate. The more common fate is galactose, just like fructose, is phosphorylated by, obviously we will say ATP to ADP will occur, by what we say to be, as I have already mentioned before, galactokinase, or very good, hexokinase. So galactose will be phosphorylated to form, just like fructose, galactose 1-phosphate, just like fructose formed fructose 1-phosphate. Okay. From here onwards is where the speciality of galactose as a metabolite lies. From here onwards things will be a little different, so bear with me. See, in the meantime there is another thing which will come, another molecule will come, and the name of that molecule is UDP glucose. What is UDP glucose? We'll talk about this later. In carbohydrate metabolism only, we'll talk about this later, I promise. UDP glucose, this is a molecule. Just for the time being, know that this is a molecule. It will come. What will happen is there will be an interchange or exchange rather of groups. Galactose 1-phosphate and UDP glucose, what will happen is there will be an exchange of groups. So this will turn into UDP galactose, okay, and this will turn into glucose 1-phosphate. So there is an exchange of groups occurring here, okay, UDP glucose to UDP galactose, and from galactose 1-phosphate to glucose 1-phosphate. Okay. What is UDP glucose, how that is coming, we'll talk about all that later in carbohydrate metabolism discussion only. Okay. By the way, the name of this enzyme here, it's a little big, but it's not illogical totally. What is that? See, galactose 1-phosphate, uridyl transferase, because there is a transfer of groups here. Transferase. Galactose 1-phosphate uridyl transferase. This is the name of the enzyme here. Okay, and this will convert UDP glucose into UDP galactose, galactose 1-phosphate into glucose 1-phosphate. Okay. Now what will happen to UDP galactose, that we are not studying right now, because glucose 1-phosphate is a major thing that will now keep on going on rather. So glucose 1-phosphate, this will be converted into, very conveniently, glucose 6-phosphate. So what will be the name of the enzyme here? Common sense. What is the name of the enzyme which does these kinds of things? We have done this thousands of times before we have done this. 1 to 6. Very good. Mutase. So this is a mutase, and what is the mutase used here? Phosphoglucomutase. Okay, phosphoglucomutase. This is the name of the enzyme which will actually facilitate glucose 1-phosphate to glucose 6-phosphate conversion. Okay, now you have glucose 6-phosphate, so you have reached one of the glycolysis intermediates, because in glycolysis of glucose also we see glucose to glucose 6-phosphate. So you have formed glucose 6-phosphate, so from here onwards what? Glycolysis, no problem. And from glucose 6-phosphate, another thing which may occur is, although this is a little less common, but a phosphatase. The main function of phosphatase enzymes is removal of phosphate. So a phosphatase enzyme may work here, and glucose 6-phosphate may be converted back to glucose. Okay, so this is one of the processes, one of the pathways by which glucose may be formed by galactose. Okay, or rather from galactose, right? So glucose 6-phosphate by phosphatase glucose. This is also one of the things, but that's not the major thing here. The major thing is that glucose 6-phosphate may go into glycolysis. Okay, and hence this is the speciality of galactose metabolism. This is galactose metabolism. Okay, so now we will obviously see, as priorly promised, as to what the speciality, or rather what the clinical relevance of the galactose metabolism is. See, as I have already mentioned when I was talking about fructose the fructose metabolism clinical relevance, that two of the enzymes there in fructose metabolism, if they were deficient, they led to two clinical disorders, right? Just like that, just like fructose metabolism, here also, if firstly beta galactosidase is deficient, we get the very common thing which I have already talked about, lactose intolerance, right? Now, if galactokinase is deficient, then we get a condition which is known as non-classical galactosemia. Whereas if the next enzyme, which is galactose 1-phosphate uridyl transferase, this enzyme is deficient, we get classical galactosemia. So let's very quickly talk about these conditions. So the first enzyme which we talked about was beta galactosidase. We have already talked about lactose intolerance. Second one's deficiency, that is galactokinase deficiency, leads to what we say to be non-classical galactosemia. Non-classical galactosemia. Now, non-classical galactosemia, this means galactose plus blood, right? So galactose in blood, galactosemia. But obviously galactose will be present in blood. Then what's the disorder here? Excess galactose. Okay, excess galactose. So when does this occur? Obviously, as I have mentioned, galactokinase deficiency, then it will occur. Now, very similar to sorbitol accumulation, listen to me very carefully. This is important. This is very important. Very you may say similar to the sorbitol accumulation leading to cataract. In non-classical galactosemia, this is occurring in non-classical galactosemia, what happens is, if there is an accumulation of galactitol, remember that alcoholic form of galactose, remember that? So if there is non-classical galactosemia, then obviously there is a large amount of galactose. Then obviously a large amount of galactose can be channeled to galactitol. Now try and remember, if there was a large amount of glucose, it could have been channeled to form sorbitol. If there is a large amount of galactose, it can be channeled to form galactitol. Both are alcoholic forms. And both sorbitol and galactitol tend to accumulate in the worst of places. That is, for example, in eye lens. So here also cataract may occur. In fact, may occur is an understatement, since cataract is the major clinical symptom here. Very important. Okay, cataract is the major clinical symptom in case of non-classical galactosemia. Okay, it's a major, and how that is happening? Due to accumulation of galactitol. Why that accumulated? Because galactose, that concentration rose. And cataract may also occur, as I've already priorly said, due to sorbitol accumulation, due to glucose concentration being high. So very similar, it is very similar. Okay. So I think these are the two things which are the most defining elements of non-classical galactosemia. Now let's talk about the classical galactosemia. Okay. Now the first thing which I would like to mention here is that the classical galactosemia is more common. It is much more common than non-classical. As mentioned in the names, classical galactosemia and that's non-classical. Okay. So this is the more common of the two. And obviously, as I have mentioned before, how does classical galactosemia occur? Why does it occur rather? Occurs due to enzyme deficiency. Which enzyme deficiency? As I have said, it was galactose 1-phosphate uridyl transferase, right? The deficiency of that. So galactose 1-phosphate uridyl transferase, its deficiency will lead to classical galactosemia. Okay. Now what actually happens in this is this is classical galactosemia is, by the way, as I've already mentioned, that since these are enzymatic problems, they are autosomal recessive. This is also autosomal recessive. Okay. So classical galactosemia is also an autosomal recessive disorder. So what are the symptoms of classical galactosemia? Classical galactosemia, the first symptom and the most important symptom in classical galactosemia is jaundice. Very, very important. Jaundice is the first sign. Okay, it is the first sign in classical galactosemia. Jaundice was another symptom in which of the disorders that we talked about? Yes, very good. Hereditary fructose intolerance. In hereditary fructose intolerance also, jaundice was a symptom because there was rapid hepatocytes death. Okay. Here also the liver is very vastly affected. How? Jaundice, firstly, hepatomegaly is another brutal condition. The liver will enlarge in size, right? And cataract is also, just like non-classical galactosemia, cataract is also a symptom. But unlike non-classical galactosemia, cataract is not the primary symptom. Here the primary symptom is jaundice, whereas in non-classical galactosemia the primary condition or symptom was, yes, cataract. Okay, although cataract occurs in this, or rather has a chance of occurring in classical galactosemia also. Okay, next is mental retardation. Mental retardation also may occur, right? And we obviously have physical symptoms like vomiting, seizures, etc. Right? So these are kind of the symptoms which we see. But the most important is obviously jaundice, because that's the first sign. And if this is jaundice, you can link it to liver anomalies. So hepatomegaly linked to this, and cataract, since this is, you're talking about galactosemia here, and just like non-classical galactosemia, cataract may also occur. Mental retardation obviously occurs, and vomiting is another physical symptom which classical galactosemia presents with. So what is the treatment here? The treatment is not very, I would say, medical or something like that. It's rather preventing this. There is no actual treatment, there is much more like preventing this. And the prevention of this is you remove galactose sources. So removal of galactose and lactose from the diet, that will kind of treat this, or rather at least help in dealing with these conditions. right? So with this, we actually complete galactose metabolism. We completed galactose metabolism, we completed fructose metabolism, we completed the clinical relevance of the galactose fructose metabolisms, and we also talked about the sorbitol pathway, and we talked about how the sorbitol pathway leads to cataracts in diabetics, which is an extremely important clinical aspect. In this discussion, we will not be able to cover the gluconeogenesis part as I had hoped to cover, but it's already past 1 hour already. So I think it would be too much. So we'll talk about that in the next discussion, which will be coming out very soon. And in the next discussion, I think we'll be talking about both gluconeogenesis, and we'll also talk about glycogenolysis, right? And then we will move on to HMP shunt and the other aspects of carbohydrate metabolism. In the meantime, we'll also be continuing with the protein metabolism, okay? And we may even start lipid metabolism, who knows? So thank you for watching. Hope you understood, hope you got something out of the time you invested in this. Thank you.